We'll discuss now limitations of immunofluorescence. And immunofluorescence has many limitations. For example, photobleaching. What is it? It is fading of signal of immunofluorescence of a fluorophore molecule with more time and with repeated exposure to the source of light. So as we see, we want to see the cells for more time, for longer period of time, or with the repeated exposures, what happens? The signal of immunofluorescence that gets weaker and weaker. This is known as photobleaching. How we can avoid it? It can be avoided by brief exposure to the source of light. So हम मिनट्स की बजाय हाफ मिनट या उससे थोड़े टाइम के लिए एक्सपोज करें उसको. Similarly, by reducing the intensity of light, हम ज़्यादा इंटेंसिटी की लाइट जो है उसकी बजाय हम लो इंटेंसिटी की लाइट को यूज़ करें. एंड सिमिलरली by increasing the concentration of fluorophores, हम फ्लोरोफोर की जो कंसंट्रेशन है उसको बढ़ा के, जाहिर है ज़्यादा फ्लोरोफोर होगा तो उसको फोटो ब्लीच होने में ज़्यादा टाइम लगेगा. सो सिमिलरली by using reversed fluorophores or by using more efficient, more good fluorophores, जैसे हम पहले भी हमने बात की कि जो एलेक्सा फ्लोर डाइज होती हैं वो आमतौर पे कम फोटो ब्लीच होती हैं. तो एलेक्सा फ्लोर डाइज जो है उनको हम यूज करके फोटो ब्लीचिंग का जो इफेक्ट है उसको मिनिमाइज कर सकते हैं. तो दिस इज फर्स्ट लिमिटेशन ऑफ फोटो ब्लीचिंग. Similarly, there is fixation of cells. We have also discussed that when we use antibodies, which are labeled with some fluorophore for seeing the cells, these are big molecules. These cannot pass through the cell membrane of the cells. So what we have to do, we treat the cells in such a way that the cells, they are no longer in living state. So ये भी एक limitation है कि हम सेल्स के जो डिफरेंट इवेंट्स होते हैं उनको हम डायरेक्टली लिविंग स्टेट में, लिविंग फॉर्म में स्टडी नहीं कर सकते हैं. सो हम जो भी सेल्स में स्टडी करते हैं वो उनके वो इवेंट्स होते हैं, वो फेजेस होती हैं जो वो नॉन लिविंग फॉर्म में होती हैं. सो दिस इज अनदर लिमिटेशन ऑफ द immunofluorescence. Similarly, there is cross reactivity. What is cross reactivity? It's primary or secondary antibodies which we use in our studies. These may bind with some other molecules present in the cytoplasm or on the plasma membrane. So when they bind with other molecules, they do not bind with their respective antigens, then we get false positive results. And this, of course, is cross reactivity. But we can, instead of using these primary labeled antibodies and secondary labeled antibodies, we have a solution to it. And what is it? We can use recombinant proteins. For example, this GFP or some other proteins. What is GFP? Green. This is green fluorescent protein. These may be transfected in the cells, and when these are expressed, the cells will have natural fluorophore present in their cytoplasm or in some other organelles which express these proteins. So these proteins like GFP or others, they will have no photobleaching, they will have no cross-reactivity, as we have discussed. Similarly, we can see the cells, we can study the cells in the living form. So instead of seeing the dead cells or fixed cells, we are able to see the living cells. So जो इसके जो main limitations हैं, इसमें photo bleaching है, इसमें fixed cells जो ज़ाहिर है हम during treatment living cells नहीं रहते, वो हमने discuss किए, हमने देखा कि cross reactivity भी होती है. So these are different limitations of immunofluorescence.